KMD Bioscience provides quality assurance for antibody humanization services, including mice, rats, rabbits, etc. Humanized antibody is an antibody from a non-human species that has been modified to increase its similarity to antibodies produced naturally in humans. Humanization monoclonal antibody refers to tailoring non-human monoclonals to work within human immune systems to minimize immunogenicity while increasing therapeutic efficacy by including human antibody sequences into existing non-human frameworks. Chimeric antibodies. These antibodies are produced by combining the variable domains of a murine antibody, which are responsible for antigen binding, with the constant domains of a human antibody. Chimeric antibodies are thus part human and part mouse, with approximately 70% of the antibody being human. This modification significantly reduces their immunogenicity compared to fully murine antibodies, but still can trigger immune responses because of the murine antigen binding regions. Humanized antibodies. In contrast to chimeric antibodies, humanized antibodies are designed to be even more similar to human antibodies. Only the CDRs of the murine antibody are grafted onto a human antibody framework. As a result, humanized antibodies consist of a greater proportion of human sequences, over 90%, further reducing the likelihood of immune responses when administered to patients. Selection of parental antibody. Identify a non-human antibody with high specificity and affinity for the target antigen. Screening and selection based on antigen binding characteristics. Characterization of the parental antibody. Understand the structure and binding properties of the antibody. Sequencing the antibody genes, determining the crystal structure of the antigen-antibody complex, if possible, and identifying the complementarity determining regions, CDRs, and framework regions, FRs. Selection of human framework. Choose a human antibody framework that is compatible with the non-human CDRs. Process. Database search for human antibodies with similar framework sequences and structural compatibility with the parental antibody. CDR grafting. Graft the non-human CDRs onto the selected human antibody framework. Synthesize genes encoding the chimeric variable regions, human framework plus non-human CDRs, and clone them into an expression vector. Molecular modeling and optimization. Ensure the grafted CDRs adopt the correct conformation for antigen binding. Use computational modeling to predict the structure of the humanized antibody and identify any framework residues that might need modification to support the structure of the CDRs. Synthesis and expression of humanized antibody. Produce the humanized antibody in a suitable host system. Transfect mammalian cells, e.g., CHO cells, with the expression vector containing the humanized antibody genes, followed by culturing and purification of the antibody. Verification of antigen binding affinity and specificity. Confirm that the humanized antibody retains the antigen binding characteristics of the parental antibody. ELISA, surface plasmon resonance, SPR, or other binding assays to compare the affinity and specificity of the humanized antibody against the target antigen with that of the parental antibody. Optimization and back mutation. If necessary, optimize the performance of the humanized antibody. Introduce back mutations, selective reversion of certain framework residues to their original non-human sequences, or other modifications based on structural and functional analysis to improve antigen binding affinity or antibody stability. Humanized antibodies have broad applications in the treatment of various diseases, especially in oncology, autoimmune diseases, and infectious diseases. Their reduced immunogenicity compared to murine and chimeric antibodies makes them particularly valuable for chronic treatments where long-term administration is required. Some notable applications include oncology. Humanized antibodies such as trastuzumab, Herceptin, target HER2 in breast cancer, improving outcomes for patients with HER2-positive breast cancer. Another example is alemtuzumab, which targets CD52 in lymphocytes and is used in the treatment of chronic lymphocytic leukemia. Autoimmune diseases. Humanized antibodies like daclizumab, which targets the IL-2 receptor on activated T cells, have been used in the treatment of multiple sclerosis, reducing the frequency of relapses. Infectious diseases. Palivizumab, a humanized antibody targeting the RSV virus, is used to prevent RSV infection in high-risk infants, demonstrating the utility of humanized antibodies beyond cancer and autoimmune diseases. Other therapeutic areas. The development of humanized antibodies continues to expand into other therapeutic areas, including cardiovascular diseases, transplant rejection, and allergic diseases, showcasing their versatility and potential in modern medicine. Collectively, KMD Bioscience antibody humanization services provide transformative solutions for optimizing the therapeutic potential of antibodies, ultimately helping to develop safer and more effective treatments. Whether you are a pharmaceutical company or a research institution, our expertise in humanized antibodies can drive your project to success.